Cassie, Marketing Specialist from GenScript, and we are the world's leading biotech company with gene synthesis, protein, antibody, and preclinical drug development service capabilities. So in this webinar today, we're going to discuss about SARS-CoV-2 neutralization antibody test for the evaluation of neutralization antibody prevalence in Singapore. So let me introduce our panelists today. We have Dr. Leong who is the infectious disease physician from Mount Elizabeth Novena Hospital, Professor Wang Linfa from Infectious Disease Program in Duke NUS Medical School, and lastly, Dr. Leong, who is the field application scientist from GenScript. Dr. Leong and Prof. Wang are both experts in infectious disease, and they'll be discussing about their experience in historic emerging zoonotic infectious diseases, challenges in COVID-19 management, limitations of existing serology methods, and why neutralizing antibody detection can add value in COVID-19 management. We will also review the novel cPass SARS-CoV-2 neutralization antibody detection kit from GenScript in terms of its performance evaluation, the information that the cPass test can provide, and antibody longevity characteristic for binding versus neutralizing antibodies. This webinar will take around 50 minutes and it will be a recorded session. You can type in your questions in the chat box and we will answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll be sure to take down and I will email back to you. So without further ado, let's welcome Dr. Leong to start his presentation. So Dr. Leong, over to you right now.
Hi, okay, I think I'm sharing screen now.
Yes.
Okay, great. Okay, now I'm going to talk a little bit about antibodies and neutralizing antibodies. This is fascinating because as you realize, antibodies can form to any part of the virus structure. And what you really want to do when you actually control a virus is actually have neutralizing antibodies. And you realize at the brink of it all in the whole epidemic is actually the vaccines. The vaccines can actually help us turn the tide against our fight for COVID-19. Can neutralizing antibodies help us? We're going to talk a little bit about this. So first of all, I'm going to string this. Antibodies, and we realize that the antibodies, we always tend to think that antibodies are very specific to the antigens. It's not always true, but more than 90% of the time it's true, and we kind of assume that this is it. But false positives do occur, and you realize it in your role as a doctor. You've seen false positive HIV screening tests, etc. So nothing is perfect in this world. Certainly antibodies sometimes have false positives as well. And you also realize that the virus is not just about the spike protein. There are many other proteins to it. And you have the nuclear antigens, the surface proteins, the core proteins, and what we call NS and the non-structural proteins. You've come across the dengue NS1 antigen. So the NS1 stands for non-structural protein, the first one. And there's also the polymerase gene, which will sometimes come as POL. So what happens is that the body, as it gets attacked by the virus, all these will develop, and these are antigens to them, and the body can mount a different response and different antibodies to them. So these are the 4 antigens which are drawn up. Then the body can actually mount up specific antibodies to all of them. Now, not all of them will work to block the virus. It's just a response recognizing it's foreign, but what you really want to know is which of it neutralizes it. Because once you identify a neutralizing antibody, there's role for vaccines, there's role for therapeutics. And I'm going to go a little bit about that later. So antibodies are developed against different antigens. Just an example, in hepatitis B, you've learned about the surface antigen, which in turn gives you a surface antibody. You have the core antigen, which gives you a core antibody, but you don't use it similarly. If you want to look for previous infection, you actually do the core antibody. If you want to look for immunity against hepatitis B, you look for the anti-hepatitis B surface antibody. So different antibody has different purposes. Now, Epstein-Barr virus is even more complicated. You have the VCA, which is a viral capsid antigen, the EA, which is the early antigen, and the EBNA, which is the Epstein-Barr nuclear antigen. So I often get calls from GPs asking me, what test do I do? So for acute infections, you actually use the VCA antibodies. So this is going to be a little bit confusing. Acute infections, you do the VCA IgM to diagnose it, but again, there are risks of false positives, and there's also risk of false negatives. So there are limitations in all these tests. Now, if you want me to ask, you have questions about the Epstein-Barr virus, contact me later after the talk, and we can talk a little bit more about this. So the role of different antibodies are to predict different things. If you're very curious, you can read this journal. It's available online freely. There's a QR code, and it tells you the different antibodies there are and what they're used for and how they differentiate different conditions. But the discussion of this is outside the talk for today. Neutralizing antibodies, then what are they? It is an antibody that's capable of keeping an infectious agent from infecting a cell by neutralizing or inhibiting its biological effect. In other words, it is almost like an antiviral thing. It stops the virus from infecting the cell. Very important, very helpful. If you actually look at all the different viruses we have, not all of them is capable of developing neutralizing antibodies. A very good example that's very effective is Hep A and B. The vaccines, you know which antibodies are the ones, you can make them effectively. Influenza, there are actually neutralizing antibodies to influenza, but the trouble is the virus keeps mutating, so it makes it a little bit difficult to form neutralizing antibodies. Right on the other extreme, you have hepatitis C and HIV, where the virus keeps mutating and keeps shifting, so you don't really have a good neutralizing antibodies for Hep C and HIV. But in HIV, there's something called broadly neutralizing antibodies, which is a subject that's quite different. Again, but these are ways in which people are trying to develop antibodies to help in controlling and even curing HIV. Again, different viruses have different abilities to cause neutralizing antibodies, but there is an important reason. If you know it can cause a neutralizing body, you can instigate it, you can even find it. It becomes an opportunity to develop a vaccine. It's an opportunity to develop hyperimmune antibodies so that it can actually give a rescue for the patient as a therapeutic. I'm going to show you a little bit of other data. Neutralizing antibodies in SARS, for example, the more severe patients actually have higher amount of antibodies, and this have a role as protects the individual. This is actually a Nature review paper back in 2009. This is a long time ago, but look at it. It actually identified the spike protein of SARS-CoV as the target for vaccine and therapeutic development. This is very important. With this information, we were thus able to quickly identify the spike protein in COVID-19, develop vaccines and therapeutics for it. Without this foreboding, without the SARS in 2003, we wouldn't have been able to understand so much of COVID-19. So in this paper, it tells us that when you identify spike proteins, you can develop neutralizing antibodies, you can actually stimulate appropriate T-cell responses, and these are good vaccines and therapeutic options. H1N1, 2009 influenza, we can also have neutralizing antibodies against it. And this is just a paper to show you that they've been able to identify one neutralizing antibody against influenza viruses. But it is very specific. It is against this H1N1. If there's a mutation, it forms a H3N2, it is not helpful. This is yet another paper published in 2010 showing you can develop neutralizing antibodies to the pandemic influenza H1N1 as well as 1918. Again, neutralizing antibodies occur, and the best thing is they occur naturally. And are we already using plasma therapy even before COVID-19? The answer is yes. We've been making use of these neutralizing antibodies to try to treat certain conditions. Hepatitis B immunoglobulins, the doctors who are much my age or older will remember giving it to children where the mother was born with Hep B surface E antigen positive or with the surface antigen positive. So there's a typo here, is antigen positive. So these are antibodies that are given to the newborns to try to control them. CMV hyperimmune antibodies are also given. We use it in the control of CMV disease in transplant, and it has a possible role in a prevention of congenital CMV disease in pregnant mothers who are exposed to primary CMV. There's some good data coming from there. So neutralizing antibodies, if you find them, you can make them, or you can actually get it from a pooled plasma from the blood donation. And in turn, there are therapeutic options. Now, coming back to COVID-19, role of neutralizing antibodies, I'm going to go through a little bit about the whole history of COVID-19. Let's say the exposure happened here at the beginning of the arrow. Then thereafter, we have the onset of symptoms, and this usually happens about 5 days later, up to 14 days later. And you do know that the PCR turns positive just before the onset of symptoms. And the PCR may remain positive for weeks to months. Certainly, we have reports of people who are positive on PCR for 3 to 4 months, but usually it's about 4 weeks to 1 month. Now, very importantly, on day 14 to day 21, the antibodies start developing. Your serology starts turning positive. This is a very, very important timeline because when you have a positive serology, it tells you that the infection occurred at least day 14 to 21 beyond. If your PCR is positive, it tells you these are the likely times in which the infection could have occurred, and with the serology, you can time it. And the serology is very helpful. For some reason, I think it's rather fortuitous as that on day 40, it turns positive. And we know that infectivity of the individual to transmit the virus is in the first 10 to 14 days. The Germans showed that you can't transmit after the 10 days, but in Singapore, it showed that we can still transmit up to the 13th or 14th day. So come 14th day of illness, you can't transmit the virus, which means when the serology turns positive, you can't transmit. And it makes sense because the antibodies can neutralize it. So serology actually helps us very much in the management. We can determine the age of the illness. Now, and again, non-infective rates, when the serology turns positive, it tells us the person is no longer infective. Now, this is a very busy slide. I'm going to try to focus on a few things, look at where my pointer goes. Okay, the first one is about antibody and assay types. There are many, many different tests in the market, each with their own limitations. Each can cause false positives and false negatives. Then I'm going to look at the results. If you look at the results here, one thing you can see is that the trend upwards. The more days there are from the onset of illness, the more likely you're going to have a positive serology test result. So all these helps. But what I want to highlight to you is that all these tests are not 100% specific. You have a few cases where you end up with false positives, and the false positive rates are about 2%. Now that's worrying because if I'm going to do a screening, you end up with 100 cases of 100 individuals, and then 2 of them may have a false positive result. That will complicate matter, doesn't it? If you want to look at serology passport, antibody passport, this 2% may have not been infected, but in turn, they can actually cause trouble. So what I really want is a better test. Instead of all that's available in the market previously, is there a better test which you can have available? The current problems with the current serology test are that false positives occur, and 2% is too much for me. It's how long does it persist for? The antibodies, does it persist for life? Is it for 6 months, 3 months? We don't really know yet. Does this positive serology means it confers protection? Is it neutralizing? The short answer is all the tests in the markets have not been neutralizing. So all the tests you should have come across in the past until now have been non-neutralizing and doesn't confer protection. It probably is just another antibody to another part of the virus and not to the spike protein blocking the infection. And honestly, with all the serological tests, false positive PCRs, it is awfully confusing. Can we do better? And all of you have seen the fly-by-night kits. You can even find them on Cool10 or whatever websites there are. You can buy them, drop a drop of blood and see it. I can tell you it has caused a lot of frustration and angst. We've seen the kits, we've seen results from the kit, and end up with false positive results. What it means is we need a better test with something which is more certainty. In fact, can this test actually guide us in terms of the immunity of a person? Now, this is taken from a Bloomberg Business Week, and I find that it really helps me in summarizing the problems which we have with COVID-19 serology testing. This press, Stephanie Baker, this press person went on to do 4 antibody tests. At the end of the day, she's not sure if she really had COVID-19. She had a URT-like symptoms earlier on, somewhere in February and March, and she wants to know if she really had COVID-19. And none of the tests, some of the tests came out positive, some of the tests came out negative, and she's none the wiser. This is a picture taken from Bloomberg Business Week, and it just shows her frustration. What do we want? We actually just need a better antibody test. We need a better kit. Again, just a few reminders. Antibodies are not all equal. Some are neutralizing, some are non-neutralizing. And depending on which antibody you use, you will ask different questions and you get different answers, and it will have help you in your management in a different way. Different antibodies provide different information. Use the right antibody test to get your answers. Neutralizing antibodies, very exciting, because it's how we're going to develop vaccines, and this is how we're going to develop therapeutics. And of course, better diagnostics. Can neutralizing antibodies help us in our vaccine assessment? So what better diagnostics do we have against the COVID-19? Okay, I'm going to stop the talk here and pass the talk to Professor Wang, someone who has been who's literally the Batman of the world, who has been leading the charge against COVID-19 as well as SARS back in 2003. It is a great pleasure to share this speaking time with him. Thank you, Professor Wang.
Yeah, thank you, Dr. Leong, for the great presentation. So right now we'll just invite Professor Wang Linfa to introduce the surrogate virus neutralization test technology. Professor Wang, over to you.
Okay, can you see my screening?
Yes, we can.
Okay, first of all, thank you, Dr. Leong. I mean, what an introduction talk. You know, you set the bar so high, but I think I have to say, you know, that's why we need you to start, because the clinical relevance and the challenge, I think you laid out, you know, just crystal clear. So it's my great pleasure. You know, I have been in Singapore for 8 years, and I came here really try to be prepared for pandemics. And, you know, it's unfortunate for the society to have it, but as a scientist, you know, I feel like very, very privileged to be able to contribute and in Singapore. So really, to continue the talk, obviously, this is all about neutralizing antibodies. And so what we're going to try to do is, you know, in Singapore first, but I would say that, you know, the impact will go way beyond Singapore and in the region. So the talk will be 3 parts. And so I will be focused on the technology, and Dr. Lee from GenScript will be describing the commercial kit and also the ordering process. Now, I think, you know, every one of you, you really, you know, have seen these numbers every day in and day out, and you have seen this, you know, the map. As Dr. Leong says, you know, I've been in this game for 30 years, and I was involved in SARS, and before that was in Nipah, before that was in the hantavirus in Australia. I think the COVID-19 pandemic really made 2 things, even for scientists like myself, really unexpected and very, very sort of unusual, right? So the first one is you look at the map, you know, it started in China, in Wuhan. Now you look at this, that dot is not dark at all, and it's really limited. Instead, you know, you have these developed nations, USA, Europe, you know, and it's really, really dark. So that's the first. Secondly is that in the last 30 years, I have been involved in 6 emerging zoonotic outbreaks from Hangzhou, SARS, MERS, Ebola, to COVID-19. And COVID-19 is different because the containment failed. So we now need an exit strategy. So that's why vaccine, neutralizing antibody, all going to play a much more important role in this pandemic than the previous one. So in terms of diagnostic tests, Prof. Leong Dr. Leong already says, there are many, not only antibody tests, but also a PCR antigen test. So it's kind of interesting. We give you a time frame of the infection. So early infection is that PCR, and now Singapore government is adopting this RAT, the rapid antigen test. So this you go after the pathogen, basically, either the nucleic acid or the antigen. And then once you get to the convalescent stage, then we go for antibodies. And then most of the tests so far, actually all the tests commercially available, can only detect binding antibody. And the one we developed, the surrogate virus neutralization, is the only one now commercially available, but actually directly really go into, assess the neutralizing antibody. So this sort of arrow indicates the time course of the infection. But I personally think it's also like, it's an indicator of the importance of this test in the pandemic, evolution. Early on, really, diagnosis is the most important. We want to see whether people are infected or not, and we do contact tracing and we do containment. But now, evolved to today, and especially now the vaccine is our market. So we're talking about exit strategy, whether it's a prevalence, the antibody, or the immunity, longevity of immunity, and then the immunity passport. All of these rely on the accurate measurement of neutralizing antibodies. So the significance of measuring neutralizing antibodies, I think, have emphasized enough by Dr. Leong. So I'm going to go very, very quickly. The vaccine efficacy, right? So Dr. Leong says not all antibodies are made equal. I'm going to just add on, not all vaccines are made equal. So obviously, and also not all individuals who receive the vaccine will be equal. So take a vaccine is good, but there's no guarantee that you have the neutralizing antibody or you are protected. So of course, in terms of protection, we have cellular immunity and also the functional immune response, neutralizing antibody and the binding antibodies. I always say, I'll be the first one to admit that neutralizing antibody is not the only arm of protection. T-cell immunity is always very important. But on the practicality level, especially for GPs in the audience today, you are never going to be able to prescribe a T-cell assay because it's just not going to happen, because it will never be commercially available, and the FDA or HSA will never approve that because it's a research test, not a routine clinic test. And then in terms of therapy, whether you use convalescent antibody or monoclonal antibodies, right now, for example, plasma therapy, even in Singapore and the USA, I think it's used largely blindly. The doctor make a decision to say maybe plasma will work, but with a very preliminary trials in Singapore, we already found the cases that the recipient already have a higher neutralizing antibody than the plasma you're going to give to them. The reason is that we don't have an easy way to determine neutralizing antibody and in a clinical setting. So this, the cPass, the surrogate virus neutralization will change that as well. So currently, the gold standard of measuring neutralizing antibodies, regardless of which virus, whether it's COVID-19, HIV, Ebola virus, or Nipah virus, you have to use the live virus. And this is what we call a gold standard, a virus neutralization test. And so how the test works is the virus will bind the receptor in the cell. And for COVID-19, it's BSS3 containment. For Ebola and Nipah, it's BSS4 containment, like in Singapore, which could not do it. So the live virus binds the cell and that's that infection. If you have neutralizing antibody there, it will block the virus enter, and so you neutralize the infection. The advantage is specificity. This is the most specific test, but there are many, many disadvantages. It requires biosafety containment. It's very tedious and slow, and you need a really highly trained staff, and it will never be commercially available, because the real cost per test will be $500,000, if you put all the overhead onto it. And you need people like that, you need to really protect your gear and go to the BSS3. And the readout is like that, so you need a highly skilled trained person. So in late February, early March, I've been following this, very closely. I already felt there's a need for this. So we had a concept of, can we really replace that with a much simpler assay we can do in a clinic setting, even in a GP clinic. So we term this a surrogate virus neutralization. So on the left, this is the live virus neutralization in the virus in the cell. The surrogate virus neutralization actually is very simple in principle, but it's 25 years of basic research. Especially the last 15 years from SARS-1, we learned all about the receptor, ACE2 is important, and also the spike protein, and also the receptor binding domain. So basically, it's through protein engineering, we invented about chemical simulation of the virus-host interaction. And then in the presence of neutralizing antibody, basically, we see a signal reduction. So without the neutralizing antibody, the HRP conjugate RBD will bind to the ACE2 in an ELISA plate, just a common ELISA test, basically, a blocking test. So now what we can contrast is that the current live virus neutralization that everybody else is doing still require BSS3 in the live virus and days of waiting. With what we have here now is a surrogate virus neutralization. It can be used in a BSS2 or clinical lab, very fast and can be automated, and high throughput, no issue, much more consistent because the variation is less than 5%, interplay, and interplay is less than 10%. Whereas the virus neutralization can easily be 50% difference, And then the stop, we have a SOP that global harmonization is easy. So this is in comparison with the live virus neutralization test. In comparison with all the current commercialized tests, and I have been told the FDA already approved like 50, and commercially there's close to 200 antibody tests. All of them are doing binding tests. And so far, nobody can claim that they can measure neutralizing antibody. So the sVNT really is the only one now commercially available and FDA approved. Okay, so we designed really specifically to test for neutralizing antibodies, and then we can confirm the protect function and does not need any secondary antibody, only one wash step. The last is important, it's isotype independent. Most of these antibody tests on the left, they detect IgG, IgM, or IgA. And we know that the immune response in individuals are different. Like recently, I've been told a vaccinee took a Moderna vaccine and had IgM antibody, but no IgG and the neutralization negative. So our test is isotype independent. We really ask, does the individual have neutralizing antibody, and we don't care whether it's IgG, IgM, or IgA, but mostly it's IgG, of course. And the other point is equally important, but for doctors in the audience today, you guys may be less concerned, but the origin of the virus, right? It's still a very hot topic. WHO just organized a mission, and I was on the mission 17 years ago and led to the team discover the origin of SARS. What you need is to use the neutralizing antibodies, but capable of performance in a species independent. So our test has been already applied for 14 different animals, and I always say any animal serial you have, I can test for you. So why neutralizing antibody is important? Because that whether you look at the binding antibodies or T-cell immune responses, these are even not an exhaustive list. We have more data now. So you look at the cellular responses, whether it's CD4, CD8, you can see almost all the proteins that are encoded by the COVID-19 virus, they have a response. And for binding antibodies, again, most of the antigens that they have a response. But for neutralizing antibodies, we know that really only come from the spike protein. On the right, you can see is that, so in a human serial, if you bind to the RBD, then you say, is that just, as long as you bind to the receptor binding domain, are they all neutralizing? No. Even if you bind to the most critical antigen, you still have two types of binding antibody versus neutralizing antibody. So this is a figure came from our Nature Biotech paper. So you can answer these questions using monoclonal antibodies, because monoclonal antibodies only bind to one epitope or one site on the protein. So all these monoclonal antibodies binds to RBD. So 1, 2, 3, 4, as you see color-coded. And then you come to the right, as you can see that only 2 out of 4 actually neutralizing, so only 50% of success. That's why it's so important that you have to develop a test which actually really measure the neutralizing antibody rather than by correlation or deduction, okay? Of course, once you have a test like that, people want to see data, and also people want to see data not only from your own lab or your collaborator, they want independent verification. So there are many publications already. Our sVNT test has been used over 30 countries now in research and some in clinical settings. And so I just give you two examples. One is from a Malik group in Hong Kong University. He is internationally well known and worked on SARS, and now is a leader in COVID-19. So they had 47 positive and 107 negative. And I don't want to have the time to go into really the detailed data, but the conclusion is really important. What they did is using this sera panel, both positive and negative, and compare with the classic virus neutralization we call plaque reduction neutralization test. So this is the one that you have to do it in BSS3 and wait for 3 to 5 days. But the end result is important. Both in sensitivity and specificity in this cohort, they achieve 100%. And then there's another test done in Europe. This is exactly the same. You have a PNT and you have an inhibition with our sVNT. Again, highly, highly concordant. Then, of course, as a scientist, I don't want to really criticize other people's tests, but I think to be fair is that we try to do correlations, right? So what we have is ABC, 3 different commercial tests, the front runners of the other antibody tests, which does not measure neutralizing antibodies. So now if you look at this, you use the binding antibody and you try to look at the neutralization, now the correlation drops, right? The R square is 0.29 to all the way to 0.4, the middle sort of a 0.5 region, but you never get to the 0.8, 0.9. And here is another study, uses much more sample and a different test. As you can see that, the binding antibodies, the blue one, the curve is, we rank this from weak to strong. And then these are the neutralizing antibodies. You can say there's a weak correlation, but definitely not good enough as a clinical test. The other really relative new information came out in the last few months that I did not have the knowledge, like when I developed this surrogate virus neutralization in March, April. This information came out from maybe July. So you look at the longevity of antibodies, and then to our surprise, or maybe I should not be surprised, is that our immune system has a way really to select for the most important functional antibody, that's the neutralizing antibody. So neutralizing antibody is almost selected over the binding antibody. As you can see that, the binding antibodies went faster than the neutralizing antibody. So this is one Nature Medicine, and this is now in meta-archive, so it's not a peer review yet, but the trend is there, and our own data also suggests that. So I think it's scientifically already accepted as a fact. Of course, individually you can still have variation, but at a population level, the trend is really very, very, accepted. And then, what we have also is in our region, like the MH in Malaysia, already use our sVNT and to do this, and also get really good 100% sensitivity to do with RT-PCR. I think what I'm going to raise is really this Ethos lab in USA. This is something that I think is very impressive. So this is a private lab. They were searching on the market for commercial tests to do neutralization, and they discovered our test. And they actually went ahead and offered a commercial service for testing neutralizing antibody. Now that's very brave. In the context of so many competing sort of tests on the market. And the other binding antibody tests already got FDA approval, whereas ours, at that time, we still did not have FDA approval, but they went ahead, offered the neutralization test, and now they have customers from all these phase 3 clinical trials, vaccine trials, go to there, and they want to know the neutralizing antibody, because this is the only test that they can do. All the other tests only tell you binding antibodies. Okay, so a very, very important question that everybody's asking, and can antibody test predict protective immunity? one thing is clear, Dr. Leong already says, and I have presented all the data to say binding antibodies measured by all the commercial tests right now we have does not equal to neutralizing antibody. So that's clear. Now we have a test to measure neutralizing antibody. So the question is, does neutralizing antibodies equal to or protect immunity? We will say most likely. So this is like, most, Peter Dowdy, the Nobel laureate, says that, but I think the face of the pandemic, Tony Fauci says that as well. I mean, he is on the news almost every day. He gets so excited with the vaccine trials. So this, the quote came from phase 1 trial. Of course, now we know phase 3, the Moderna vaccine is 90–95% effective, at least to prevent disease. But even early, in phase 1, Tony basically says, I'm really excited because these produce high neutralizing antibodies, and it's a good assumption that neutralizing antibody will be a good correlate of immunity, protect immunity. But these are all comments. Let's show you some of the scientific data. So 3 papers I'm going to cite among the many published. The first one is the most interesting. So this is the study done by the University of Washington in Seattle. So what they did is to follow a crew of fishing vessels. So the fishing crew that they go out to the sea to do fishing. And before they went out, 144 of them, they took their blood, they did a PCR. All of them are PCR negative, but in terms of the antibody test, they have 6 are positive using a commercial binding antibody, and 3 are positive using our neutralization test. Months later, when they came back, it's incredible because it's in a enclosed containment. 85% attack rate. 85 of the 144 got infected with SARS coronavirus, and including these 3 which were positive in the binding antibody but negative in our neutralizing antibody. But the 3 which are positive in our neutralizing antibody were fully protected. None of them get an infection. So that was the first indication to say neutralizing antibodies correlate with protection. Of course, this is a small cohort. And then in the last few weeks, there are many papers coming out. So this one is not accepted yet on the right. It's done by Oxford University. Basically, they went to sample zeros really on the regular basis, and over 10,000, 13,000 of them frontline healthcare workers, and they just monitor their serum samples longitudinally, regularly, and then they look at the people who got infection. And again, they conclude that the neutralizing antibody is sufficient really to provide protection. This paper just came out last week in Nature. So this is the most conclusive now. It's a passive protection experiment. So they use neutralizing antibodies from immunized monkeys and then transfer them to naive monkeys. And the conclusion is that neutralizing antibody is protective in a dose-dependent fashion. And T cells will also play a role, but only when the neutralizing antibody is very, very low, okay? And that also that you don't need much of the antibodies, that actually relatively low level of neutralizing antibody is sufficient to protect. Okay, so, as I said, as a scientist who have worked in this field and now 8 years in Singapore, obviously I'm very, very proud of being able to develop these tests very rapidly. I always say it's COVID-19 speed. We had the concept in February. We had the preliminary data in March. We filed the patent in March. We worked with DXD and the GenScript, and we have the product launched in May, and now we have FDA approval. So it's a very, very pleasant, teamwork, and I thank, of course, DXD and the GenScript for that. And in terms of how we can use sVNT and neutralizing antibody, I think, everything from now on, the exit strategy will rely on neutralizing antibody, and also that retrospectively try to go back to look for the origin of SARS-CoV-2 and the intermediate host of SARS-CoV-2. Also for pandemic preparedness, now we have a neutralizing test. We can actually prospectively do monitoring and surveillance in animal and human populations. So if a related virus pop up, before they cause a large pandemic, we can also do early warning. So the application is, unlimited. But for today's kind of a forum, of course, clinical relevance, of course, the vaccine, the therapeutics, and also interestingly is that if you want to find the new drugs, this is also a high throughput test. And actually, some of the research lab is already using this to screen for new novel compounds that which can block the IBD. And so I will finish up with this, a provocative thought for the doctors in the audience, and if there's a policymakers in the audience as well. I still hold an Australian passport, but I'm not allowed to travel back to Australia. So I thought this news on November 23rd. Qantas basically says, we need approval of COVID-19 vaccine before you can travel to Australia as an international passenger. So I was asking myself, is a piece of vaccine certificate going to be enough? Because even with the most effective vaccine, as Tony Fauci was claiming, is 95% effective in preventing disease, okay? We still don't know what's the percentage effectiveness of preventing infection or preventing spread. As Dr. Leong just says, even if you have 2%, false negative, or if that protection 10 or 20% is not protected against infection or spread, then we're still at risk until the herd immunity reaches 70–80%. So in the early phase of vaccination, I think a piece of paper saying you have been vaccinated may not be enough. And of course, I'm biased, but I think most likely we need to see also the neutralizing antibody level to make sure really you're protected. With that, I'd like to thank you, and I'm going to pass this to Dr. Lee to do the last two parts. Dr. Lee.
Thank you, Prof.
Yes.
Wang.
Right, so let's invite Dr. Lee from GenScript to introduce the cPass SARS-CoV-2 neutralization antibody detection kit. Dr. Lee, over to you right now.
Yeah, can you see my screen now?
Just now we can, but not now. Maybe you can try to share again. Yes, we can see it.
Many thanks to Prof. Wang to have shared the lighthouse of the various applications of the sVNT technology. Huge pleasure for me to speak after the two celebrity clinician and scientist. My name is Yan Feng. So I'm the field application scientist representing your technical support from GenScript. I'll host the last 5 minutes of the presentation to introduce the cPass IVD kits that were fomented and developed from the sVNT technology. Lastly, followed by how can clinicians get access to order the cPass test. So on November 6th, the US FDA authorized the first serology test that detects neutralizing antibody from recent or prior SARS-CoV-2 infection. In this cPass news release, right, FDA also explains the neutralizing antibodies that can bind to a specific part of the pathogen and may be responsible to decrease the SARS-CoV-2 viral infection of the cells. If you can still vividly remember, the 100% sensitivity specificity between cPass compared to the PRNT from the independent validation from Hong Kong and also Germany. So likewise, we have produced cPass performance data using PRNT as comparator for the review by FDA. So with this sample set of 26 positive and 88 negative, as you can see from the top table on clinical agreement using PRNT 50 titers as the comparator methods. All 26 positive samples tested using PRNT 50 was found positive in cPass, and all 88 negative samples were negative as tested in cPass. So PRNT 50 is actually indicating the serum titer that can produce 50% of plaque reduction. Same consistent conclusion using the more stringent PRNT 90, as you can see from the bottom table. So FDA is actually recommending GenScript to continue evolving the cPass technology to allow much more features and also broader applications. Stay tuned and we will definitely update you for more applications. Now, I would like to bring your attention back to our homeland, Singapore, as HSA provisional authorized cPass neutralized antibody detection kit will be used locally for COVID-19 detection. cPass is a blood test assessing neutralizing antibodies post-infection, which is the closest biomarker to breach with one's immunity. The cPass actually will offer two unique information from one test. Number one, determine who has neutralizing antibody. Number two, who has been previously infected. So in this slide, please find the cPass performance evaluation using RT-PCR as comparator. Total 47 positive samples and 931 negative samples have been run in the study. All positive samples were collected at least 2 weeks post-infection. As you can see, there is 100% sensitivity for all 47 positive samples and also 99% specificity for negative samples. In all, cPass can deliver who has neutralizing antibody and who has been previously infected with good sensitivity and specificity. So now we're coming to the last agenda for the presentation. If you're equally convinced of the cPass clinical utility, you may wonder, how do I order the cPass test? So in this slide, the GPs in the audience may have already received this circular and probably familiar with the MOH guidelines on use of COVID-19 serology tests. So if a serology test is performed for any of the reasons, including contact tracing, epidemiology study, or any of diagnostic testings, patients with first known positive serology test, including cPass, will be issued MC based on whether patient is asymptomatic or showing COVID-19 symptoms. So for individual diagnostic testing or population-level serum surveillance studies, cPass can deliver both information who have been previously infected, presence of neutralizing antibodies. So the clinicians might wonder, who should get tested for neutralizing antibodies? So we listed a few for your references. Number one, frontline workers or individuals who may have higher exposure to the COVID-19. And some of these individuals may be asymptomatic but have already developed neutralizing antibodies against COVID-19. So other individuals who may be asymptomatic but do not have clinical indications to COVID-19 can also test for neutralizing antibodies. Individuals who may require serology test results to demonstrate the status of a previous infection. Lastly but not least, individuals who would like to investigate possible false negative COVID-19 results and past infection. So many of the examples can be available for doctors to consider. This is not an exhausted list. So like any serology testing procedure, cPass test can be done with blood sampling, laboratory analysis, and report receiving. So we are in talks with the Parkway Health Laboratory regarding the distribution of this test, and they will update you once it becomes available for ordering. So thanks for your attention. And now I would like to invite back Dr. Leong, Prof. Wang, and Parkway team to join me to address your questions. Thank you so much for the audience.
Thank you, Dr. Lee, for the presentation. So now we'll answer some of the questions from the floor. So the first question, do we know how long does neutralizing antibodies last in the body after a COVID-19 infection? And is there any difference between symptomatic and asymptomatic infection? Maybe Prof. Wang, do you want to take the question?
I'll go first, and Dr. Leong will add on. So we have a paper right now reviewing the Lancet, so that I can share because I think this is publicly important information. With the cohort that in Singapore that we have done, around 200, without any doubt, the second question is easy to answer, that the neutralizing antibody correlates with the severity of disease, and Dr. Leong already said that. If you have more severe, you have a high neutralizing antibody. The second question is being debated in the media, even in the scientific community. First, they say, oh, neutralizing antibody only lasts 3 months. And then, the Iceland paper say, oh, you can go 4 months and 6 months. What we have done is much deeper. We look at the avidity, neutralizing antibody, sVNT, everything. And we have even a machine learning mathematical modeling. So our title is that to say the neutralizing antibodies can vary from 39 days. We have one patient, the half-life is only like 39 days, and just months later it's gone. In the other extreme, we have patients that the neutralizing antibody did not change for 6 months, even going up. So using our method modeling, that person, the neutralizing antibody can last 39 years. Okay, so this is the spectrum. Basically, I always say that for COVID-19, like a pandemic, I think it's impossible to say that it will not last or it will last forever for everybody. But that's why, again, it's so important that for individuals to check it out. Yeah, Dr. Leong?
Prof,
Oh, sorry, Dr.
yeah, okay, got it.
Right now.
So I have nothing else to add. In fact, but I really find it very interesting that the neutralization antibody, you can actually see what's the level, and that will in turn predict the risk of you getting infections. So if I'm the Australian government, I'm not going to say do a COVID vaccine, because honestly, there are so many vaccines out there in the market, and so many of them has so much variability. In fact, what I would rather do if I'm the Australian government is show me a cPass test. Show me a cPass test with good antibodies before you fly. Perhaps if you're positive on that, we're not going to quantify and say you're going to last 39 years or 39 days. But if you're positive on that, in the last, then for next 1 month, you're considered as safe. Because if anything, the presence of neutralizing antibodies will mean you are protected at least for a certain period of time. I think that's a better correlation than having had the vaccines. And you heard it, even in Modena, 195 of them, 5 of them will not have, will not be protected. That's because I suspect they are negative on cPass. That means it's not a pass but a fail.
Yeah, thank you. I cannot reveal the individuals, but we already have the phase 3 clinical trials that people have IgM but no neutralizing antibody. The other thing, Dr. Leong, is important is in high prevalence country like Europe and the USA, again, it's more important to have the neutralizing antibody certificate, because from past infections, you may have something better than the vaccine, So yeah, agree. Yeah, but I'm biased.
Okay, thank you, Prof. Wang and Dr. Leong for the answers. So the next question from the floor, does different strains of COVID-19 produce different types of neutralizing antibodies?
I'll leave it to Prof to answer.
Yeah, I will have a go at that. So far, this is a much more difficult question to answer, because the nature is that we do neutralizing antibody determination retrospective, right? As Dr. Leong says already in the convalescent stage. So unless you do a control experiment that you have the genome type of that person when in the first 10 days, you miss that opportunity. All you know is that person has neutralizing antibody. So if you see a subtle difference of that neutralizing antibody, you could not conclude because they infect with a slight different virus. So what we have done is the reverse. What we have done is to grow, like in Singapore, we have 13 different genome type of virus we already isolate in the Duke and Ye's lab. So we have 13 different viruses, including the famous, the D641G, 614G, and the G clade, the L clade, it's in the media. So when we use this, and then you use patient sera, we don't care whether they infect with G clade or L clade or the D or the G. Then if you do that to this different virus for neutralization, then you see subtle difference for some strain and mostly no difference. So I think the overall consensus right now is to say, yes, we see genetic variants in the virus, but so far all the variants will have such a insignificant difference. It means that like the cPass will be able to detect all neutralizing antibodies with 90–95% reliability, and also no matter which vaccines you use, it will be protective against the current strain. But as scientists, we never say never, right? Once you apply the vaccine, whether you force the virus to mutate to have an escape mutant, that will become less effective using the current vaccine. So I cannot predict future, but with the current knowledge, first of all, the virus neutralization testing cover all the sera, and also the vaccine should be able to protect all the strain we know now. Yeah.
Thank you, Prof. Wang, for the explanation.
Yeah.
So the next question, the cPass assay uses RBD as the target, but there are neutralizing antibodies targeting at the region outside RBD on S1 protein. Will cPass miss that portion of the neutralizing antibody?
Great. If that's a question from a doctor, that's a very, very smart doctor, because this is a very scientific question. And that's the reason our paper was rejected by Nature and we went to Nature Biotechnology, because we actually use the S1, RBD, and the whole spike protein, okay? So that definitely there are neutralizing antibodies binds outside the RBD region, but we have quantified that portion will be maximum 10%. In most patients will be less than 5%. So that's the first statement. Yes, there are S1 neutralizing antibodies, but they are less than 5 to 10%. Secondly is that if you use the S1 protein in our sVNT, the conformation makes it less sensitive, because that actually it does not get blocked by neutralizing antibody as sensitive as RBD. So we actually, if you read our paper in the original Nature Biotech paper, we tried all of the possibilities. Eventually, we landed on the RBD as a compromise, because it's much more sensitive. And yes, we may miss 5 to 10% antibody, but it's still more sensitive than if you use S1.
Thank you, Prof. Wang. So the next question, what's the evidence to address whether cPass positive individuals are still able to transmit the virus to the others? That is, does positivity protect both the individual and also the community?
That's a tough question. We don't have the data, and the data coming from all the published studies. So that especially the University of Washington, then definitely it says that these 3 individuals which are cPass positive are immune, protected against getting infection, and then by default, if you did not get infected, you could not transmit. But I have to say that we don't have data beyond that. Okay, and it's going to be a very difficult study. And Dr. Leong, you were saying that I should encourage Australian government to have a policy. I said you can help me to convince the Singapore government has a policy, is that we should do a really wide range of surveillance right now to get the cPass baseline today in December, and then do another maybe 50,000 people in January if the vaccine starts, and then another 50,000 every month. We did 50,000 with the vaccine coming on. First of all, we will differentiate between neutralizing antibody come from vaccine and the infection. So we have a test now, we can differentiate this. Secondly is that among these populations, if they go overseas to travel, then we will know whether they when they get exposed in US and other countries, whether any of these with a decent cPass rating can still get infected. I may add on as well to the doctor who asked this question is right now internationally we're still struggling with the harmonizations. The test is not harmonized, but even the standards are not harmonized. So I'm on the committee with WHO. We are hopeful that in December this month, by the end of the year, the WHO with CEPI, with the National Institute of Standards for Biological Controls, is going to release a panel of sera that they have a predefined neutralizing antibody titer, and they will call this international units. And we will use our cPass to calibrate to that international units. And hopefully by March next year, that we have enough data to say, you can use cPass to measure your neutralizing antibody, translate this to IU, and on the scale of the 1 to 10, do you need an IU of 4 and above to be protective? So these are all my dreams. We're working very closely with the international bodies. And just like a Hep B, right? When you do a test, you have IU, and the doctor now knows what IU level you have to achieve to get a protection. So we are working toward that with the COVID-19. Of course, Hep B, you had decades of experience. COVID-19, we only have 10 months, So, but as the vaccine demonstrates for COVID-19, everything can be accelerated, and we are hopeful by middle to late next year, we will have an IU, and we can advise your clients to say, do a cPass and give them IU, and then they have some confidence that if IU reach that threshold, you will be protected.
Thank you, Prof. Wang. So we do have 1 minute left, so I think we can take on the last question. The last question from the floor, does steroid therapy affect development of neutralizing antibodies?
That's what I missed the first
does
part.
steroid therapy affect the
Steroid
development?
therapy.
Yes, correct.
Dr. Leong, that's a doctor's question. You know, basically immune suppression, right?
Yes, it is immune suppression. It really depends on the degree of immunosuppression. In a say in an extreme like heme transplant situation, you may actually negate and not have any antibodies form. But in most of the steroids which are used in the practical situation and the kind of immunosuppression we're talking about, we should be able to. Again, this is a big question we should always struggle when we do patients and whether there's a mounted immune response or not. So it really depends on the gradation of immune suppression you have. And again, different people will respond differently even to the same infection, the same dose. So that is also yet a varying factor.
Yeah. And it depends on when you apply that steroid a treatment. So I love to, if somebody can get blood from Trump, I love to test him because he got this dexamethasone very, very early. I think no other patient has had that. So if you are thinking of this may suppress neutralizing antibody production, he will be the best test case.
Thank you, Dr. Leong and Prof. Wang. So due to time constraint, we will end the Q&A session over here. So for those questions that we didn't get to answer just now, I will email back the answers from you, back to you. So thank you again, Dr. Leong, Prof. Wang, and Dr. Lee for the presentation, and also to all of you who joined us today. I hope you find this session useful and beneficial for your work. So thank you, everyone, and have a nice day. Bye-bye.
Thank you. Bye. Good.